Hello, my name is Dan Tempest, and today I'm going to be talking to you about the renal system and how it regulates changes in hydrogen ion concentration. So I want to talk to you about acidosis versus alkalosis. And in order to do that, I first need to just mention that there are three regulatory systems as far as acid-base balance in the body. So the body uses buffers. It also uses the lungs or respiratory system. But in this presentation, we're going to be focusing on the kidneys. Want to point out the Henderson-Hasselbalch equation because this helps us to determine the pH of a solution. So you can see the Henderson-Hasselbalch equation upper right here, up there. So in this case, the pH is equal to the pK, which is the dissociation constant, plus the log of the concentration of base over the concentration of acid. And pH is going to be essentially as hydrogen ion concentration increases, that's going to make pH go down and become more acidic. And this would be known as acidosis, therefore. And if there's less hydrogen ion concentration, that's going to make our pH go up, making it more basic. And that's going to be called alkalosis. Just for reference, normal pH of blood in the human body is around 7.4, so slightly over neutral. Now, one really important part of acid-base balance is the bicarbonate and carbon dioxide. And this is going to be important for maintaining pH. So this bicarbonate is really regulated by the kidneys. Now, the kidneys are actually quite slow to respond to changes in pH, but their response is the strongest of the three regulatory systems. So, in metabolic acidosis, so in this case, we have hydrogen ion concentration that is high or increasing. That hydrogen ion will then be secreted into the nephron to be excreted. In that same example of metabolic acidosis, if bicarbonate is low, it will be produced more and then reabsorbed into the blood. So all that's going to begin happening at the kidney. Opposite of that, if we have metabolic alkalosis, so in this case, if hydrogen ion concentration is low, the secretion is going to be slowed. And if bicarbonate levels are therefore high, less of this is going to be produced. So we're going to go through a couple examples of how this might occur. So first, we have the type A intercalated cells. This is going to be mostly associated with acidosis. So the type A intercalated cell, we're going to have hydrogen ion secretion, but it's going to be coupled with bicarbonate reabsorption. So just to point out a little bit of where that would be found, here we have bicarbonate, I should probably pick a different color. Bicarbonate reabsorption is going to be up here, and that's going to be reabsorbed into the bloodstream. And then on the opposite side, here we have the hydrogen ion secretion into the tubular lumen. So again, hydrogen ion secretion is going to be coupled with that bicarbonate reabsorption. A couple things to note. First, we have carbon dioxide entering the cell, and it's going to combine with water, and that's going to form carbonic acid, which will dissociate into our bicarbonate ion and the hydrogen ions that we're talking about. The hydrogen ion is then actively transported into the tubular lumen. We see this actively transported because here we have these ATPs. These ATPs, energy is required to do this active transport. This is actually known as primary active transport. It is important to also point out that this is done by an enzyme ATPase, and it is also coupled with potassium ion entering the cell. And here we have that potassium ion entering the cell. Bicarbonate, again, is reabsorbed into the bloodstream. That is done in exchange for chloride ion. So we see that right here. Okay, so that's also coupled. The hydrogen ion secretion will then lower the pH of the urine, and this change in pH is going to be limited by some buffers such as bicarbonate and also phosphate buffers, which I'll talk a little bit about later. This can create carbonic acid, which can then form carbon dioxide and water that can actually be reabsorbed into the cell. So that's the type A intercalated cell. Again, mostly associated with acidosis. So when the levels of hydrogen ions are high in the blood. Okay. Opposite of that, if we look at type B cells, the type B intercalated cells are going to work opposite, again, to the type A ones. In this case, we're going to be secreting bicarbonate back into the tubular lumen. We see that right here. And that's going to be in exchange for the chloride ion. Point that out. Here's our chloride ion. And hydrogen then is going to be reabsorbed into the blood. And this is exactly opposite. You see that right here. And that's going to be in exchange for the potassium ion. Again, this is more associated with alkalosis. So when there's lower levels of hydrogen ion found. So we need to increase that. Again, all to work with homeostasis of keeping the pH or the acid-base balance of the system. All right. To kind of summarize this up now. Oh, also the phosphate buffer. I just want to talk about that real quickly. The phosphate buffer is going to help to control the drops in urinary pH. The phosphate buffer is going to take on the excess hydrogen ion concentration, and it's going to become a titratable acid. And that titratable acid will be excreted in the urine. We find that right here. So here's our phosphate buffer, and here we have the excess hydrogen ion combining with that buffer to form this titratable acid again that is excreted in that urine. It is important to also point out that this process also forms new bicarbonate. Okay, so that's pretty important as well. And that can be found, that's going to be reabsorbed into the bloodstream. So again, phosphate buffer, another pretty important part of maintaining that acid-base balance in the kidneys. All right, just to kind of bring it all back together, here we have bicarbonate and hydrogen ion regulation as a whole. We have hydrogen ion secretion over here on the left side of the diagram, and bicarbonate being reabsorbed. So when hydrogen ion concentration in the blood is going to be high, so that would be here. When that is high, the type A intercalated cells, we're going to have more hydrogen get excreted. That leads us to this step in the urine. That's going to be through the process of acid or because of acidosis. And on the opposite side, and that's going to bring down plasma pH, so bring down that pH. On the opposite side, when hydrogen ion levels are lower in concentration in the blood, the kidneys are going to reduce that secretion by type A cells, but they're also going to increase the activity of the type B intercalated cells, which are going to excrete more bicarbonate. And that would bring up the pH. So the pH comes back up. All this, again, feedback loops. In this case, pH of the blood got a little too high. We're going to have acidosis. And in order to get that back, we need to excrete hydrogen ion. Opposite side, if the pH gets too low, it gets too high, pH gets too high, we're going to excrete bicarbonate, bring it back down. pH gets too low, we need to bring it back up. pH gets too high, we need to bring it back down. Okay, all the negative feedback loop of the homeostatic mechanisms of acid-base balance. All right. So now I got into a journal, and in this particular journal, essentially we have different groups of rabbits, and just to give it a little background, the alkali-rich diets are often recommended in human medicine to prevent the pathological consequences of nutrient acid load. So if you want to avoid acid, we can manipulate that a little bit with base-rich diets, essentially, or also known as alkali-rich diets. And that's going to help with people who have impaired renal function or impaired kidney function, essentially. This study, and essentially what we had rabbits, and that's going to be our lab animal in this case. And they were exposed to differing levels of alkali intake, essentially. And what we did is we tracked acid-base balance, but also renal control in this herbivore. Okay, so in this case, we had only male rabbits, and they were kept in a metabolism cage. Their urine content and arterial blood samples were then analyzed for data, and we'll analyze that data here in a little bit. The metabolic carbon dioxide production was also measured, and that was to help calculate alveolar ventilation. And there are 3 randomized groups in this experiment. And they were fed variable levels of alkali diet. Okay. So if we look at this first figure, one thing you'll notice that as renal base excretion, so more base is being excreted by the kidneys, that is related to dietary alkali load. Okay, so if we look here, we have 3 groups. Here are our 3 groups. We have high alkali food in the solid dark. So, and you'll notice that for the majority of that's going to be found up here on our graph. Then we have the normal alkali food, and then those are going to be mostly found in this range. And then we have the low alkali food, which are almost exclusively found down in this range. Okay. So that was when we compared, again, dietary alkali load. So how much alkali food they had in their diet. The higher alkali food they have, so the more basic food they have, the more base excretion we had. That would make sense, right? If we're feeding these rabbits more alkali food, we would expect more to be excreted. And if they have the normal, it was more in the mid-range. And if they had really low alkali food, there's not as much to be excreted. So again, this made sense. If we look over at the right graph, we were looking at base reabsorption here. So how much base is being reabsorbed versus the dietary alkali load? Again, those 3 groups. So again, if we analyze this a little bit, and that low alkali food, most of that base is being reabsorbed, and that would make sense because they're not getting as much base in their diet. They need to reabsorb most of that base. If we look at that normal group, again, they fall right in the middle. And if we look at the high, we have a larger group over here. And again, that would make sense. As they're eating more base, they don't need to reabsorb as much because they're getting a lot of that in their diet. All right, so just to go through some results and conclusions to this data, the normal and high alkali chow in the urine, we had the bicarbonate was excreted daily, and that was typically containing insoluble precipitates of about 35 to 60% carbonate. And on that low alkali diet, the mean pH decreased all the way down to 6.25. In comparison, in that high and normal alkali chow, a pH of about 8, so quite a bit lower. And again, that would make sense. They have more basic food, so they're going to be excreting more basic urine. In conclusion, the herbivore nutritional alkali load, so how much food base they're getting in their food, elicited large rates of renal base excretion. So more alkali load, more food alkali in the food, they're going to get more base excretion. Again, that would make sense. To which the urinary tract of the rabbit appeared to be adapted to handle that load. So they were able to get rid of it, in other words. So a strongly base deficient diet exerted maximum impact on the renal base saving mechanisms, implying that there is already a condition for growing susceptibility of metabolic acidosis. So if we look at this image here, again, when the dietary alkali load is analyzed, we have the high, the normal, and the low, and we see huge differences here in the distribution. Very high amounts of base being excreted, medium, and very low. If we look at renal base excretion again, very high, also very high, but very, very low in the low. However, if we look at the systemic buffer base, we see a pretty standard across the board. Not a whole lot of scientific significance between those samples there. Okay, so again, essentially what we figured out in this experiment is that if you feed rabbits a very high alkaline diet, they're going to excrete more base, and if you feed them low, they're going to try and reabsorb, which again would make sense. And here are my references, and I hope you enjoyed my presentation. Take care.